So this is one of my favorite chapters, chapter 16, and it's about cell signaling. I just think cell signaling is the heart, the meat and potatoes of modern cell biology. Understanding cell signaling really helps us to understand a lot of physiology and physiologic effects of drugs and cell signaling, and understanding of cell signaling is really where all the research is right now. So in this video, we're going to talk about the general principles of cell signaling in very general terms, and then we'll get specific with how G protein coupled receptors work and a couple of examples of that, a couple of enzyme coupled receptors. So let's get started. So some of the general principles of cell signaling. You've probably heard of the endocrine system, and in general, endocrine means that the signal that's released by the signaling cell is going to travel via the bloodstream to distant targets. Its specificity comes from, as in all cell signaling, the presence of the receptor on the target cell. So endocrine signaling means that the signaling molecule, whatever that happens to be, hormone or whatever, is traveling via the bloodstream to very distant targets. In contrast, paracrine signaling is not going to use the bloodstream. It's going to use just Brownian motion to travel through the interstitial spaces and target cells that are very close by. That's what para means, is next to, and so paracrine signaling is going to be targeting very close by cells. A very specific kind of paracrine signaling that we've already talked about is the neuronal signaling. So if you understand neuronal signaling, you'll understand paracrine signaling, because the endocrine cell in this case is the neuron, and it is releasing a signal that travels to a very close by cell. In this case, the target cell is only 40 nanometers away across the synapse. So most paracrine signaling a little bit further distance than in neuronal signaling, but neuronal signaling is a specific kind of paracrine signaling. There's an even closer paracrine signaling, and that's autocrine, which is not on this slide, but autocrine would be if the cell that is releasing the signal also has the receptor for the signal. And this is really common in feedback mechanisms in neurons. For instance, all of the cannabinoids are released by neurons for self-modulation. The cannabinoid receptor happens to be on the same cell that's releasing the cannabinoid. And then the last kind of signaling would be contact dependent. So something like a notch delta, where one cell has the notch, the other cell has the delta, and this is really good for setting up borders, things like patterning in stripes or coloration, or the edges of tissues like an organ's barriers or the side of the organ. So one cell says, I'm a notch cell, and the other cell says, well, then I'm a delta cell, and the notch and delta bind to each other, and so the contact dependent signaling is the last kind of signaling. So so that's one way we break up signaling. Another is by excuse me. Another is by the chemical nature of these signals. So we've got lots of different kinds of signals: steroid-derived, protein-derived, some that are derived from peptides or amino acids, like, for instance, adrenaline is derived from tyrosine. Many of the hormones that you think of as hormones, like estrogen and testosterone, are all derived from cholesterol, so those would be steroid hormones. Insulin, which we've talked about before, is a protein, and so it's a gene product. But it can even be like small molecules. For instance, nitric oxide as a dissolved gas acts as a signaling molecule. So that's another way that we can divide up signaling, is by the chemical nature of the signal. So how the signal gets to its target is one way, and then the chemical nature is another way that we often divide up cell signaling. And another is by the receptor type. So receptor types increase the diversity of the signaling that could go on downstream of the release of the signal. For instance, when you release adrenaline, there are alpha receptors on one kind of smooth muscle in the bloodstream, and beta receptors on another kind. And so what's going to happen in response to adrenaline is really going to depend on the receptor type. An example of that is here, acetylcholine on muscle cells, on heart muscle cells, is going to bind to a metabotropic acetylcholine receptor, a muscarinic receptor, and it's going to cause the relaxation of the cardiac muscle. So that's very different than when acetylcholine binds to the ionotropic receptor on smooth muscle or skeletal muscle. So in that case, we would have acetylcholine is going to cause contraction of the muscle, and that's what you see at neuromuscular junctions. And a third result of acetylcholine is like in the salivary glands, the receptor, metabotropic receptor again, but here it's a different tissue type. And so it results in the secretion of saliva. So here we see that the metabotropic receptor in two different cell types has different downstream consequences. Or in the same cell type, two muscle cells, by having different receptors, the acetylcholine can have different effects. So that's going to increase our diversity of effect that we can get from any one signaling molecule. So a limited set of signaling molecules can have a wider array of responses by having different receptors. And then another way that we increase the number of effects is by having multiple signaling pathways, and then having an integration of these pathways. So, for instance, you could have several signals working together to tell the cell, just do your job, but those same signals and a couple more might tell the cell to divide, and then some different signals in addition might tell the cell to differentiate, and then the lack of all of these signals might tell the cell to go into apoptosis. So integration of multiple signaling pathways is another way we can get diversity of action. So a good example of this is like the patterning in your hand. So you've got notch delta signaling there, but you've also got wingless gradients across your hand from thumb to pinky, and then you've got hedgehog gradients from back of the hand to the front of the hand, and all of those pathways working together is how you end up with the very complex structure of the hand, which cell is going to be a pinky and which cell is going to be a thumb. So integration is another way that we can get a lot of diversity. So in graduate school we had this one like killer question that Jim Rand used to like to ask, and that was if a signal happens in or if a signaling event happens and then the effect happens within seconds, what do you think was the signaling mechanism? If it happens within minutes, what do you think was the signaling mechanism? And if it happens within hours, what do you think was the signaling mechanism? And so, in general, if you're opening a channel, then the signaling mechanism is going to be within seconds. You definitely saw this in the neurons. Opening a channel caused the action potential very, very quickly, right? But then, if it happens within minutes, you're probably talking about some kind of enzyme activity. So a metabotropic receptor that requires the turning on of an enzyme to make an intracellular signaling cascade kick off. Well, that's going to take minutes. And then if the signaling cascade requires new gene products, so some sort of altered DNA synthesis, so transcriptional control, and then you're talking much longer, 30 minutes, an hour, several hours. And then if it takes weeks, like in the case of serotonin reuptake inhibitors, they don't have action for like 3, 4, 5 weeks. Then you're talking about wholesale restructuring of tissue, and that's going to take longer. So signaling can be really rapid, or signaling can be slow and delayed. And this is also a good thing. You know, like when you have that fight or flight, it takes minutes to recover from that. You might still be feeling the effects of that adrenaline for 20 or 30 minutes, and that will keep you in this heightened state of awareness. Of course, long-term stress can have other consequences, and that's that hours and weeks kind of restructuring of tissue. Another interesting thing about signaling is remember that there were the steroid-derived signals, and then there were the small molecules or peptide derived, and that is going to affect not only what pathway you're affecting, but also where the receptor can be. So if it's a protein like insulin, then the receptor is going to have to be on the cell surface, because that peptide is not going to be able to be moved into the cell. So you're going to have a regulated release, which we saw in insulin, and then the downstream cell is going to have the receptor on the surface. In contrast, hormones, steroid hormones, the steroids can go across the cell membrane very easily. We know that because cholesterol is in the membrane, isn't it? And so things like insulin and testosterone, they can't have a regulated release where you make a bunch of them, put them in a vesicle, and wait for the signal to release them, because they'll just drift right across that membrane. And so you can't release them, and you can't pre-make them. And so to release them, you have to make them right then. Likewise, the receptor has to be on the inside of the cell, because all of these cholesterol-derived hormones are going to just drift right across the membrane, and so their receptor is going to have to be internal. They're going to have an internal receptor which changes shape, and that change in shape changes like whether it can be imported into the nucleus for transcription, for instance. And so the nature of the signal is going to affect where the receptor is and how we release it. And so this is a really important concept, and that's where I'll start the next video.